Hi guys, today we will be discussing our project topic, which is viral immunotherapy for melanoma. The viral therapy we will be focusing on is talomongene leperetvec, or otherwise known as T-Vec. The photo shown here is a photo taken on a cellular level showcasing the effectiveness of T-Vec. We hope you guys enjoy learning about this as much as we did. Starting with the basics of what is skin cancer. As we know, any type of cancer is uncontrollable growth of the cells. This comes in 3 types in the skin: basal cell carcinoma, squamous cell carcinoma, and melanoma. This can spread to other locations in the body. Melanoma is the most fatal of skin cancers. Fun fact, Bob Marley actually died from melanoma. Melanoma has to do with the growth of our melanocytes. Moles can be considered precancerous because they involve a lot of melanocytes, which increase the risk of melanoma. Melanoma can grow horizontally and vertically through the layers of the skin. As we stated before, melanoma is considered the most deadly because it can metastasize quickly. The tumor cell has a large nuclei and produces melanin with a variety of colors.
The causes of skin cancer can be both genetic and environmental. The leading environmental cause is UV exposure from the sun or artificial. Viral infections, immunosuppressions, and having light skin are all precursors to skin cancer. Skin cancer can present itself in a mole. To remember the worrisome signs of melanoma, one can remember the mnemonic ABCDE. Asymmetrically shaped, borders are irregular, coloration varies, diameter larger than the size of a pencil eraser, and rapidly evolving. There are 4 stages of melanoma. Stages 0 and 1, the melanoma has not grown deeper than the epidermis and it's usually treated through surgery. Stages 3 and 4, the cancer has reached the lymph nodes and possibly other areas of the body. Besides surgically removing the tumor, the patient would need some kind of therapy drug to reduce the risk of it coming back.
Melanoma is diagnosed with a tissue biopsy, and treatment can include removal, radiation, chemotherapy, or immunotherapy. Let's take a closer look on how immunotherapy works.
Immunotherapy increases the immune system. T cells police the body against infections. Usually the body will identify abnormal cells, but melanoma disguises itself. Immunotherapy will identify these melanoma cells and bust their disguise, allowing the immune system to fight against what is destroying the body. Immunotherapy is usually given through an IV drip and can be effective to a variety of cancers.
A new emerging and promising immunotherapy is oncolytic viruses. These viruses can be found in nature or created in a laboratory. The only FDA-approved virus is known as T-Vec, which is a genetically altered version of herpes. These viruses are inserted directly into a tumor, which causes the cancer cells to burst, which then leads to a secondary response. The response allows the immune system to redirect responses to other cancer cells, lysing them. Now taking a closer look at talomongene leperetvec. The melanoma cancer cell appears, and our immune system comes to combat it, which results in a protective protein shield formed by the tumor to block the immune response. When T-Vec is injected, it allows the protective protein shield to be broken down. This allows the immune system to attack the cancer cell. This can decrease the amount of cancer cells in some patients, and in others completely demolish it. Okay, so we talked to you about how there is an altered herpes virus called T-Vec that can be a new and emerging way to target melanoma. But how exactly does this work on a cellular level? Professors of medicine and oncology and hematology at the University of Alabama came together to conduct and publish research on T-Vec. Published on the U.S. National Library of Medicine, the professors discussed the biology of T-Vec. Oncolytic viruses showcase 2 different responses to a tumor. The first being the ability to be inserted directly into a tumor, selectively replicating and lysing the tumor cell. The other being a systematic immune response. Their ability to do this is because of the altered genome of the virus. The virus was originally a type 1 herpes simplex virus, which was originally isolated from a cold sore. The double-stranded DNA is extremely lytic, meaning destructive, and can cause infection and recurrent blisters in times of stress. This infection and blister gene was removed and altered to create T-Vec. 2 specific parts of the genome, ICP34.5 and ICP47, were removed to prevent the typical herpes virus symptoms to occur in a patient receiving T-Vec. Now let's talk about the cellular mechanism that makes T-Vec effective towards combating melanoma. The virus enters the tumor through a glycoprotein that helps facilitate intracellular adhesion called surface nectins. Once the virus is in the cell, it preferably disrupts the mutated protein kinase receptor and interferon pathway. When these pathways don't run smoothly, an accumulation of mutations occurs and cancer is formed. The PKR receptor may have a lower activity due to the overactive RAS signaling. PKR signaling regulates cell proliferation by phosphorylating EIF2, which prevents delivery of met tRNAi to ribosomes, blocking protein translation. In normal cells, PKR is activated by viral infection, specifically dsRNA that blocks protein translation. Cancer cell blockade of PKR signaling through hyperactive RAS allows propagation of T-Vec because the viral infection of T-Vec no longer activates the antiviral PKR response. T-Vec also replicates in cancer cells due to the disruption of the IFN signaling. Type 1 IFN signaling activates PKR to block protein translation and induces transcription of IFN-stimulated genes, such as several IFN-regulated factors that promote anti-tumor and antiviral responses. The disrupted IFN signaling can result in a loss of regulation in type 1 IFN receptors or blocking the JAK STAT signaling. Following the viral replication in the nucleus, the virions complete their assembly in the cytoplasm and induce the cell lysis to occur. The lysis of the cell will have a systematic effect due to the release of the intracellular contents. These can include, but are not limited to, viral-based pathogen-associated molecular patterns, otherwise known as CAMPs, the virons, damage-associated molecular pattern, otherwise known as DAMPs, and the cytokines. When all these components are released, a response is orchestrated. This is the recruitment of the antigen-presenting cells, such as dendritic cells, that can present themselves as cytotoxic T cells. The T cells, in turn, can mediate direct rejection of other tumor antigen-expressing cells. Decided in red are the factors that are directly produced by T-Vec, and in black are the factors produced as a result of the antiviral response.
Inside a healthy cell, T-Vec is unable to replicate, leaving the cell unharmed. Inside a cancer cell, T-Vec replicates and secretes granulocyte macrophage colony stimulating factor, also known as GM-CSF.
GM-CHF is a monomeric glycoprotein secreted by macrophages, T cells, and natural killer cells. It functions as a cytokine. This cytokine is a protein, and it's in a category of signaling molecules that mediates and regulates immunity. GM-CHF promotes dendritic cell accumulation at sites of inflammation and enhances antigen-presenting cell function. So now we have seen how T-Vec has two responses to combating melanoma, one being the lysis of the cell and the other being the systematic immune response due to the cytokine GM-CSF.
There was a study where 2,116 melanoma tumors were injected with T-Vec and 995 of them completely disappeared. The study also showed that 216 tumors that were not directly injected also ended up disappearing. This concludes that the change in the immune system was killing cancerous cells on its own. T-Vec is administered within a phosphate saline buffer solution. The direct volume of T-Vec and buffer solution added depends on the size and progression of the tumor. After receiving the first injection, the patient can receive their second dosage 2 to 3 weeks later. The frequency of receiving the dosages can vary whether the tumors are being destroyed or if the melanoma worsens. Patients can receive dosages every 2 weeks for at least 6 months until no injectable tumors remain or any other treatment is required. The patients that have received T-Vec therapy were able to tolerate it, and most of the side effects were minimal. T-Vec has very few serious side effects, and no treatment-related deaths have occurred. The side effects that come with T-Vec are similar to flu-like symptoms. The most common side effects that appear in over 30% of the patients receiving T-Vec are fatigue, chills, fever, and nausea. The less common side effects that appear in 10 to 20% of patients receiving T-Vec are abdominal pain, muscle joint pain, dizziness, and injection site pain. T-Vec represents the first oncolytic virus, which is a new introduction of drugs that directly kills tumor cells and induces tumor-specific immune responses. T-Vec has been approved to treat melanoma after demonstrating clinical benefits in trials and having tolerable symptoms. T-Vec can be considered in patients with accessible lesions for injection by ultrasound-guided injection and is most useful for older patients who cannot undergo surgery. T-Vec is another immunotherapy available for the treatment of melanoma.